Hello, dear students. Welcome back to GTC. In this video, we are going to discuss 20 important MCQ, which will going to cover your 360 degree of your pharma competitive exam. Let's start with the first question from this protein part. So the first question is linear sequence of amino acid resulting from the backbone of which type of protein? So you will see in our class we have discussed protein have four type of structure: primary structure, secondary structure, tertiary structure, and quaternary structure. In this four structure, which one you have to find is a linear one? This is very simple question, direct question. You can easily answer. Comment your answer in the comment section so that I will check how many of you have given the correct answer. Yes, tell me the correct answer. Linear sequence of amino acid forming the backbone of which type of protein, whether it is a primary structure, whether it is a secondary structure, whether it is a tertiary structure, or whether it is a quaternary structure. Yes, it is the correct answer is primary structure. If I will explain, you will understand it clearly. So primary structure is your linear one. If it is a linear sequence, then it is a primary. If it is twisted, then it is secondary. If it is twisted and forming a complicated structure, more twisted one, this is a tertiary. When this multiple chain of this tertiary structure join together, it will form a quaternary structure. So you remember question asked from this structure also. Next question. Three-dimensional structure is present. Yes, this is very easy question. Just now I have to tell you. Just now I explain you. You answer this question in the comment section. I will check how many of you have given the correct answer. Three-dimensional structure. You remember first one is linear. Second one is twisted. Third one is three-dimensional. So I told you na, this is the three-dimensional structure. If the protein is like present like this is the three-dimensional structure. When this three chain of three-dimensional structure is joined together, then it will going to form what? Then it will going to form your quaternary structure. What it will going to form? It will going to form. Okay, this is just like this. More complicated one. This will lead to form what? Your quaternary structure. If it is a single chain is there, then it is three-dimensional. If multiple three-dimensional structures join together, it will form a quaternary structure. Here, the correct answer will be tertiary structure. Next question. Identify the given structure. This is a very direct one. Which type of structure you are seeing over here? First, look at the structure. First, look at the structure. This is which type of structure? So I told you now, between these four type of structure, which structure is matching to this? Please check first, then you give your answer. Identify the given structure name. Yes. First, see the structure and comment down your answer in the comment section. If the structure is like this, then what I told it is a linear one. If it is like twisted, then I told it is a secondary. Is this structure is matching to this structure or not? Tell me, it is matching or not? This is like a twisted one, so that's why it is a secondary structure. Okay, this is a secondary structure of the protein. Next question. Identify the given structure name. A alpha helical, B beta seed, A beta helical, B alpha seed, A alpha helical, B alpha seed, and A beta helical, B beta seed. Answer this question. You have to by checking this structure. Okay, by analyzing this structure, you have to give whether it is a alpha helical beta seed or beta helical alpha seed or alpha helical beta alpha seed or beta helical beta seed. Give the correct answer. Identify the given structure. If you'll remember, let me explain you. I will give you a hint. You have to answer. So when I talk to you, its structure is quite looks like this or not? Tell me first. Its structure is quite looks like this. The twisted one. Twisted one means what? This is a secondary structure. its structure quite looks like this twisted one. Twisted one means your secondary structure. And its secondary structure is basically of two types. Okay, secondary structure of two types. That is, one is your alpha helix or alpha helical, and second one is beta plated sheet. Beta plated sheet or beta sheet. So option A is the correct answer. Alpha helical and beta sheet. This is your alpha helical. This is your beta sheet. Okay. Now moving to the next question. Myoglobin and immunoglobulin is the example of which type of protein? Primary, secondary, tertiary, or quaternary. Myoglobin and immunoglobulin. Myoglobin and immunoglobulin are which type of protein? Primary structure, secondary structure, tertiary structure, or quaternary structure. Myoglobin and immunoglobulins are example of tertiary structure. So primary structure, you remember insulin and hemoglobin B chain only. If it is a complete hemoglobin, then it will be what? If it is a complete hemoglobin, then it will be a quaternary structure. If it is a complete hemoglobin, then it is a quaternary structure. But if only beta chain is there, then it is a primary structure. Primary structure example is insulin and hemoglobin. Hemoglobin and creatinine kinase, enzyme like creatinine kinase, these are comes under your quaternary structure. Similar type of question will coming further. Okay, here the correct answer is tertiary structure. Next, choose the correct statement of quaternary structure. So you have to find which statement is correct regarding quaternary structure. Spatial arrangement, hydrophobic bonds, hemoglobin, yeah, all of them. So find out the correct statement. Choose the correct statement for quaternary structure. Yes, answer your question in the comment section. I will check which one is correct for quaternary structure. So whether it has spatial arrangement or not, yes, it has. It has a spatial arrangement of this what we can call amino acid chain. It have hydrophobic bonds. Hemoglobin is an example of this quaternary structure. That's why all of the above will be the correct answer. All points are correct regarding quaternary structure of the protein. Next question. Creatinine kinase is an example of, just now I have explained. Creatinine kinase is an example of which type of protein? Primary structure, secondary structure, tertiary structure, and quaternary structure. I want to listen the correct answer from all of you guys in the comment section. Creatinine kinase, just now I have explained. Creatinine kinase comes under which type of protein? Primary structure of the protein, secondary structure of the protein, tertiary structure of the protein, or quaternary structure of the protein. Creatinine kinase is an example of which type of protein? Primary structure, secondary structure, tertiary structure, or quaternary. Yes, just now I told you, na hemoglobin, your hemoglobin and your creatinine kinase. These two are comes under your quaternary structure. So option D is the correct answer. Next question, identify the given structure. Yes, you can see twisted one and different type of chain you can see over here. Then it is actually example of hemoglobin. Okay, it is a example of hemoglobin. So if it is a hemoglobin type example, then it will be comes under quaternary structure. Direct question now. Next question. Spatial arrangement of a protein by twisting of polypeptide chain in present in which type of spatial arrangement of protein? Twisting of polypeptide chain. Whether it is a primary, whether it is a secondary, whether it is a tertiary, or whether it is a quaternary. Give the correct answer in the comment section. Spatial arrangement. Spatial arrangement will be there if if the in the question it is there that more polypeptide chains to more than one polypeptide chain joined together due to its spatial arrangement, then it will be the correct answer in the quaternary structure. But it is not about, it has not mentioned about multiple polypeptide chain. That means it is mentioned here about single polypeptide chain. Single polypeptide chain and with a spatial arrangement means the correct answer will be secondary structure. Because I told you na, when the spatial arrangement comes to the picture, then it is basically gives a example of what? It gives a example of secondary structure. If the question is spatial arrangement of protein with more than one polypeptide chain, then it will be the correct answer will be quaternary structure. So you should uh, very clear about the what question is exactly asking. Next question. Choose the correct option from the structure of protein. You have to find the correct one. Alpha helical leucine, beta plated say histidine, tertiary structure insulin, quaternary structure hemoglobin. You have to choose the correct option. Okay. Yes. Tell me which one is the correct. Leucine, alpha helical, beta plated. Here the correct answer is you can see directly question. Quaternary structure, na? Quaternary structure with hemoglobin. So hemoglobin is an example of quaternary structure. Next question. Potassium hydroxide plus hydrated copper sulfate plus sodium potassium tartarate reagent. It is used for. That means it is asking about it is the principle of which test? Biuret test, Pauling secretu test, Heller test, yeah, Hopkins test. Yes. Who will give the correct answer? Potassium hydroxide, hydrated copper sulfate, sodium potassium tartarate. These reagents are used to in which test? Biuret, Pauling secretu, Heller's test, or Hopkins test. Read the question first. So here the correct answer is biuret test. So what happens in biuret test? Let me just briefly tell you. In case of biuret, we basically detect the peptide linkage. What we detect? We detect peptide peptide linkage. Or and second thing is at least it should have two peptide chain. Sorry, it should have two peptide linkage. It should have two peptide linkage. Okay, this is an example of peptide. Next, so here the correct answer is your biuret test. Next question. White color precipitate is observed in which type of test? White color type of precipitate. Biuret test, Pauling secretu test, Heller's test, yeah, Hopkins test. White precipitate is observed. White color precipitate. Yes. In biuret test, you will see Riemann purple color. In Pauling secretu test, you will get observe pink color. But white color precipitate you observed in case of Heller's test. Okay. Next question. Which test is identify histidine amino acid? Histidine amino acid. Yes. First test. Histidine have which heterocyclic ring? Histidine has imidazole. Excuse me. Histidine has imidazole heterocyclic ring. Then you tell me for the heterocyclic ring identification which test is used? Mollis test, Neal hydrin test, Pauli's test, or Sakaguchi test. Yes. Quickly give your answer in the comment section. Identification of histidine amino acid. Pauli's test is the correct answer. Next question. Amino acid sequence. Very important question. You have to identify the sequence of amino acid. Then which test you will be used? Sanger's test, sulfur test, Pauli's test, or Sakaguchi test. You want to detect the sequence of amino acid. Which test you will going to use? Sanger's test you will use, sulfur test, Pauli's test, Sakaguchi test. Obviously, the Sanger test, na. Sanger's reagent is basically used for the detection of amino acid sequence. So you want to check in the amino acid sequence which type of amino acid is present. Then you have to use this Sanger's reagent. Now, next question. Formation of heme in biological role of which amino acid? Formation of iron is biological role of which amino acid? Glycine, tryptophan, methionine, या aspartic acid. Okay, give the correct answer. Formation of iron in the biological sample is role of which amino acid? Glycine, tryptophan, methionine, and aspartic acid. Which amino acid play a significant role in this formation of protein? The correct answer is glycine. So glycine is the amino acid which is involved in the iron formation during the hemoglobin synthesis. Okay, hem formation, not iron, hem formation. Okay, next question. Vasodilator promotes gastric HCl and pepsin synthesis is the biological role of which amino acid? Cysteine, glutamic acid, histidine, या phenylalanine tyrosine. Vasodilation promotes gastric HCl and pepsin synthesis is a biological role of which amino acid? Cysteine amino acid, glutamic acid, histidine, या phenylalanine या tyrosine. Vasodilation promotes. Yes. So vasodilation promotes gastric acid pepsin synthesis is basically carried out by histamine. So histamine is synthesized from which amino acid? Histidine. Okay, histidine will be the correct answer. Next question. Biological role of tryptophan. Tryptophan. It is a vasoconstrictor, formation of heme, pyrimidine based synthesis, या purine based formation. Yes. What is the biological role of tryptophan? What is the biological role of tryptophan? Yes, the correct answer is vasoconstrictor. So the tryptophan amino acid is basically serves as a one of the vasoconstrictor of our body. Next, 2,2-dihydroxy indene 1,3-diol is a reagent used for Mollis test, mean hydrogen test, Pauli's test, Sakaguchi test. Very, very important question. Several times this question I've asked in exam. 2,2-dihydroxy indene 1,3-diol. This reagent is used for Mollis test, mean hydrogen test, Pauli's test, या Sakaguchi test. Why this two reagent is used? Yes, very, very important question. You will see such type of question in your exam. 2,2-dihydroxy indene 1,3-diol. This is a reagent that is used for the mean hydrogen test. Why it is used? It is used for the mean hydrogen test. Option B is the correct answer. Next question. Glycoprotein is identified by. So glyco means it has carbohydrate. Carbohydrate plus protein. So then you will detect in which test? By Mollis test, Meehan hydrogen test, Pauli's test, Sakaguchi test. Very important question and very tricky one. How you can detect a carbohydrate glycoprotein which have both carbohydrate and protein in its structure? Which test you will going to use? Whether you use Mollis test, whether you use mean hydrogen test, whether it is you use Pauli's test, or Sakaguchi test. Which test you will going to be used to detect. So if you remember in the carbohydrate class we also discussed. So carbohydrates are basically general test for carbohydrate, Mollis test I have explained you. So the same test is used for the detect. Okay, this is just a twisted one to confuse the students. So please remember the correct answer is your Mollis test. So last question of the today's video, black precipitation. Black precipitation is observed in Pauli's test, Sakaguchi test, Sanger's test, yes, sulfur test. Black precipitation is used, observed in which type of test? Pauli's test, Sakaguchi test, Sanger's test, yes, sulfur test. Yes, black color precipitate. The correct answer is sulfur test. Very good. So in case of sulfur test, we basically observed what? We basically observed black color precipitate. Okay, black precipitate. I hope you guys are understood what we have discussed. So in this video, we have discussed 20 important MCQ from the uh, different type of protein structure and color reaction part of the protein. In the next video, we will going to discuss about some important question from the disease part. What are the disease that is associated with protein metabolism? Thank you. Thank you so much. Stay connected with GDC for more such content. Thank you.